Hello, seniors. I'm glad you're still here listening to an exciting lesson related to life science. I'm Ma'am G and I will be presenting to you today's amazing lesson. So everybody, learn, focus, and have fun. For today's lesson, we will be discussing about genetic engineering. The history of genetic engineering can be traced back 1953 when James Watson discovered the structure of DNA, after which scientists were able to understand and change the structure. The idea of genetic engineering is something that we associate with the 20th century. No one knew that genes were the mechanism of heredity until the middle of 20th century. So this is our most essential learning competencies, or our MECLS. Describe the process of genetic engineering. Genetic engineering, the changing of an organism genome using biotechnology. Take note, genetic engineering is also called the genetic modification, one of the many branches of applied biology. It involves artificial manipulation or modification and recombination of DNA or deoxyribonucleic acid and other nucleic acid to alter an organism's phenotype. Genetic engineering is a process of adding a parent DNA to the genome or all genetic material of another organism. It is not natural. There are scientific intervention, meaning modify na ang genetic material ng isang organism because you want to modify something out of that organism. The target of adding one or more DNA is to have one or more traits that are not found in a certain organism. It can also be a process of inserting or transferring a gene of a specific trait into another organism. Genetic engineering is an artificial manipulation, modification, and recombination of the DNA. The term genetic engineering initially referred to various techniques used for the modification or manipulation of organisms through the process of heredity and reproduction. As such, the term embraced both artificial selection and all the interventions of biomedical techniques. Among them are artificial insemination, in vitro fertilization or test tube babies. We also have cloning and gene manipulation. In the latter part of 20th century, however, the term came to refer more specifically to methods of recombinant DNA technology or gene cloning, in which the DNA molecules from two or more sources are combined either within cells or in vitro and then inserted into host organisms in which they are able to propagate. So that one is genetic engineering. Prior in 1950s, the term gene was used to stand for the unit by which some genetic characteristics pass to generation. By 1953, we have here English chemist Francis Crick and American biologist James Watson created the DNA structure. So we have here the genetic code. Each DNA has pattern. We also have amino acids are arranged into particular sequence to produce a protein. So, yung DNA natin, we have A, T, C, G. Adenine, thymine, guanine, cytosine. Biotechnology is also important in genetic engineering or in modification of the DNA. So, ano ba yung biotechnology? Biotechnology is the use of biological techniques and engineered organisms to make products or plants and animals that have desired traits. In order to carry on this technology, it employs different processes such as, we have here the first one, restriction enzymes. So, restriction enzymes, the cutting of DNA strands into fragments. It is basically used to create DNA fragments with sticky ends or blunt ends that can join with other DNA fragments. Next is the gel electrophoresis. So, it separates DNA fragment by size. It is used to study DNA fragments of various sizes. So, that one is gel electrophoresis. We also have here the recombinant DNA technology, the combination of DNA fragment with the DNA from another source, which is also called the exogenous DNA. We also have here the gene cloning. Gene cloning is the production of the large numbers of identical recombinant DNA molecules. It is used to create large amounts of recombinant DNA to be used in genetically engineered organisms. We also have the DNA sequencing. DNA sequencing is used to identify the DNA sequence of cloned recombinant DNA molecule to further study. It is used to identify errors in the DNA sequences, to predict the function of a particular gene, and to compare two other genes with similar sequences from different organisms. So that is DNA sequencing, used to identify errors in DNA sequences. We also have polymerase chain reaction, or the PCR, makes copies of specific regions of sequence DNA. So it is used to copy DNA for any scientific investigation, including forensic analysis and also medical testing. So those are some processes under biotechnology. Aside from that, we also have the different techniques and steps in genetic engineering processes for any organisms to be modified. So we have here the first one, identification of any organisms with desirable genes. Second is the extraction of the entire DNA. Third, removal of the gene from the rest of the DNA. And the last step is the insertion of new gene to an existing organism DNA with the use of plasmid, which is a circular piece that contains the genetic material or the DNA. Okay, so those are the different genetic engineering techniques. So let us go now into the recombinant DNA technology. It is a technique of combining two DNA sequences from different sources. From the word combination, combining two DNA from different sources. One goal of this technique is cloning or producing a particular gene for analysis or mass production of a medically useful protein product. This technique uses a plasmid, usually from bacteria, as a vector or delivery vehicle of the desired trait. For example, is kapag diabetic ka, you are dependent with insulin. So, saan sila kukuha ng insulin? In order to support your needs. Meaning, they use this technology to produce insulin. So, recombinant DNA technology or they use this technology to produce insulin. Yung DNA na may trait nito, kukunin nila, mag-undergo sa recombinant DNA, tapos i-inject sa bacteria para paramihin. Dadami ang bacteria and dadami din ang trait na may insulin. So, para may insulin sila, na dedependehan kasi nga low yung insulin mo, kaya dapat ma-maintain nila yung production ng insulin. So again, ini-inject yung trait na yan sa bacteria para paramihin. Okay, and that trait that contains insulin will become more to support your needs na mga diabetic. So that one is an example of recombinant DNA technology. We have here some basic steps that involves in recombinant DNA technology. First is isolation of the gene of interest. So maa-isolate. Then preparation of vector DNA and the DNA to be cloned. Insertion of the gene to the vector molecule and ligation. Then introduction of the vector DNA to the appropriate host cell. Next is amplification of the recombinant DNA molecule in the host cell. Okay, so that one is recombinant DNA technology. Next is we have here the gene splicing. Gene splicing is the process in which fragments of DNA from one or more different microorganisms are combined to form the recombinant DNA and are made to function within the cell of a host organism. So we have here two highly significant techniques. First is the gene transfer. When we say gene transfer, transferring the gene from one source to another object. Next is the gene therapy. Gene therapy, correcting defective genes that are responsible for disease development. So we have here a sequence for gene splicing. So this one is the DNA containing desired gene removed from the cell. So this one is the cell. Yung nasa loob is the desired gene. So the DNA containing the desired gene is removed from the cell. So eto na siya. Then enzymes cut out the desired gene. So next, the enzymes insert gene into the DNA vector. This one, you insert mo dyan. Next is the bacteria reproduce, creating a large number of bacteria with the new characteristics. So you remove the desired gene, you cut it out, then insert it into the vector. Then the bacteria will reproduce, creating a large number of that new characteristics. So, ganyan lang yung gene splicing. Remove, then cut, then insert to the vector, then the bacteria will reproduce. Next, we also have here the gene therapy. Ito naman yung steps ng gene therapy. First is, the cells are removed from the patient. So, remove from the patient. Next, in the laboratory, a virus is altered so that it cannot reproduce. So, hindi na makakahawa yung virus kasi na-altered na siya, na-cut na siya. Next, a gene is inserted into the virus. 4, the altered virus is mixed with cells from the patient. So, virus plus the cell is being mixed together. Then 5, the cells from the patient become genetically altered. So, hindi na siya tinatalaban ng virus. Genetically altered na siya, okay? The altered cells are injected into the patient. Binalik yung cell dito sa patient, and take note, the cells is already altered. Hindi na tinatablan ng virus. Next is the genetically altered cells produce the desired protein or hormone. So, that is for gene therapy. Actually, this gene therapy is used in making vaccines, specifically yung sa COVID-19 natin, iba-iba yung mga vaccines natin. Gumagamit sila ng gene splicing, gumagamit sila ng vector, viral, gumagamit din sila ng gene therapy. Okay, so live virus, gene splicing, gene therapy. So, yan. So, we also have 4 approaches in gene therapy. First is a normal gene inserted to compensate for the defective gene. Abnormal gene replaced with a normal one. Abnormal gene repaired through selective reverse mutation and change the regulation of gene pairs. So, this one is again for gene therapy. A vector delivers the therapeutic gene into a patient's target cell. The target cell becomes infected with the viral vector. The vector's genetic material is inserted into the target cell. Functional proteins are created from the therapeutic gene, causing the cell to return to a normal state. So that one is a gene therapy. Aside from that, genetic engineering, we also have type of genetic modification. We have here the plant genetic modification, animal genetic modification, genetic modification of microbes. First is the plant genetic modification. Plant genetic modification is the easiest method of plant genetic modification used by our wandering ancestors, and until today is simple selection. Genetically heterogeneous population of plant is inspected, and superior individuals, plants with the most desired traits like improved palatability and yield, are selected for continued propagation. For example, is the production of pest-resistant plants, increase of crop production. We also have here animal genetic modification. Modern breeds of livestock differ dramatically from their ancestors as a result of breeding strategies. Established and emerging biotechnology in animal agriculture include assisted reproductive technologies, use of naturally occurring hormones such as recombinants, and biotechnologies to enhance reproductive efficiency without affecting the genome, and biotechnologies to enhance expression of desirable genes. Example, increase of milk production per cow. So they modify the cow in order for it to increase its milk production. Double production of eggs in chicken. So those are animal genetic modification examples. Next is the genetic modification of microbes. Microorganisms play significant roles in food production. They serve as primary and secondary roles in food fermentation and in food spoilage, and they can produce enzymes or other metabolites used in food production and processing. Humans have used the genetically modified microbes for centuries, and microorganisms are used to produce food, wine, bread, cheese, and common examples of ancient foods still popular depend on microbial ingredients and activities. Again, example, increased microbe-dependent food production and increased human immunity resistance to microbe-caused diseases. So, those are the different types of genetic modification. Again, we have plant genetic modification, animal genetic modification, and genetic modification of microbes. So that's it. That was an amazing lesson for today. Hope you learned something. Thank you for listening. See you again next discussion.